It's my pleasure to introduce you to today's speaker, Ingmar Riedel-Kruse. So I guess we start at the very beginning. When he was 10 years old, he discovered video games. He taught himself to code with an enthusiasm towards video games. And it's this type of enthusiasm you really see come up in his work later on. You fast forward, his undergraduate training was mainly was in theoretical physics, and he did his PhD in experimental biophysics at the Max Planck Institute. Now he's an assistant professor at Stanford in the Department of Bioengineering, where he has two main research focuses. The first research focus is understanding patterns of cell development and how they grow, for example, the way an embryo develops. And more interestingly is this whole human microorganism interaction area, where he is actually leading the invention of these biotic games, which allow you to control microorganisms at the very, very microscopic level and do some really amazing things. So basically, he's been able to turn a euglena into a controller where people can interact and learn about biology in a way that's really never been done before. So, please.
In the light of the whole seminar series, the main question is how can we turn microbiology into an interactive medium, right? And it started for me kind of with the idea, let's play with cells, but it's a little bit more than that now. It kind of we've broadened, and there are activities we can meaningfully do with this technology that not directly play, but are maybe playful. So let me start with something, kind of a little quiz. So what's the difference between these two pictures? The machines on the left are primarily used for number crunching, while the machines or devices on the right are really made for human experience, right? And although, they also do number crunching on a level below that. And if you look at modern biotechnology, we kind of in a similar stage now that we were back then in the late 40s, early 50s in electronics. Where, for example, here we have a microfluidic device that allows you to manipulate cells and fluids in very controlled ways. It's an integrated circuit, and you can kind of see the lines here and things. It gives you an idea of how many of those you can control. So it's pretty similar like that, but how can we convert this into something like this over the next years, decades, who knows? But these technologies get increasingly more powerful. They're used for research and also for medicine, for example. And so this technology is advancing. Will this go to something interactive like that? And for me, this started a couple of years ago, actually, when I started here at Stanford, kind of this idea, if you have this kind of power of manipulating biology at microscopic scales, why aren't we playing games with them? Or will this just develop on its own, right? And at some point I thought, maybe I should just do this, right? I should just take this on. But it was kind of intellectual curiosity, right? And if you look at the history of human technology, that's kind of a short sum up of that, you can look at each of these technologies throughout history have always been used to be playful, to interact with, and it's very prominent in our society now with video games. And it's not that clear what this means for biology, although life science is maybe the hit science coming up that is equivalent to what the information sciences or electronics have been over the past, 50 years. So the idea of biotic games then was, how do we enable a human to play with biology in a microscopic scale? And it should have something to do with modern biotechnology, right? It shouldn't be a pure electronic simulation, like spore, for example. And it also should not involve kind of like horses, which is ancient biotechnology, right? We wanted to do something novel to enable interaction. And here's one of the early examples that we made. So what you see here is parameciums. It's a single-celled organism. It's pretty large. It's 250 micrometer in size. And this is a microfluidic chamber, a very simple one, where you see the white speckles. Those are these cells. And you can apply electric fields, and these organisms have a natural response to those fields called galvanotaxis, and so they basically always swim with the field lines. And you can control these field lines with a simple controller. And you bring this in real time on a video screen and program some virtual objects on top of it. And you can make a game out of it, right? Like in this case, the goal of the game is to get these little organisms swimming through this box and score points by lighting up these dots, right? And so you can argue this is on one level, it's an interactive experiment. It's just a normal experiment. Or you can say it's an interactive, playful experience. It's a game, right? And of course, with this sort of game machine, you can make different games out of it just by programming different environments on top of it, right? So this was kind of our very early kind of approach to actually conceptualizing what does it actually mean to play with biology, right? See again, kind of the setup was rather simple at the time, which is good. It's kind of this little microfluidic chamber about 1 millimeter in size, a simple webcam, game controller, and then putting this whole thing in real time on the video screen. So we published this, got good responses, both in the more scientific as well as the geeky game literature, so kind of broad appeal. That's always good. And so this made us as a lab think we should continue on this and kind of see what can we really do with that, right? And so what next? So there are a bunch of things that we've been working on since then. And the questions fall, I mean, in technology, I mean, I didn't spend too much time on that, but the games that you saw here, I mean, you can play for 15 minutes, then you have to put new organisms in because they tire out and things like those, right? It's not the kind of experience where turn it off, play for a couple of hours, and then turn it down as we have with electronics now, right? How can we provide access? I mean, I just said that it's not that easy to use. It's even harder to give this in the hands of a child or something, right? Is the game design good, right? To what end is the game design? Is it just playing, or is there maybe educational value? Are there certain ethical considerations behind it? I mean, should we play with living organisms or not? So a number of questions, and I want to address most of them throughout my talk. Of course, there's still stuff to do, but we've gone pretty far over the past few years in that kind of area. So let me tell you a little bit about the value of, the educational value of interactive toys. One thing that fascinated me also as a child is kind of playing with Lego, and we have these Lego robotics courses, after-school program, and so forth, where kids are really enthusiastic of building these things, have these competition. And what it does is an informal way for children to learn about mechanics, mechatronics, programming. And this is really endorsed in many ways now, right? For example, the first robotics competition. Similarly, more recently, the National STEM Video Game Challenge has been announced, where children basically program video games in order to learn programming, right? And so it's not this kind of formal education. You sit in a schoolroom and you learn, or the teacher tells you to write something down. It's really like getting things done that excite you in a very playful, interactive way, right? There are some examples how games, especially in the biological context, have been used for citizen science projects to really get output from many people, so that people can really contribute to the scientific advancement. And the two examples here are folded in Eterna, where basically tens of thousands of people were able, in a virtual environment, to play around with the instantiation of real data and try to find solutions. Here, for example, in Folded, people were trying to find protein, 3D protein configurations, and were successfully in doing so, right? And you can imagine coupling these things much more directly to experimentation would be even much more powerful. And actually, Eterna, which is concerned with RNA folding, is doing that already to some extent. And in our lecture series, you will hear more about these in future talks. Another thing, just based on technology, but nothing to do with games, we can even think about other applications, for example, online education. So MOOCs have been in the news for the last couple of years. And while they are very successful in reaching many people, what has not really been solved, how can people do experiments in these kind of online classes, right? Could you couple people to do experiments, especially biology experiments? There are many scientists, for example, Albert Einstein, that have great insight, great ideas, but they have two left hands and very hard for them to do any experiment, right? Could we couple them somehow, enable them to do experiments? And there's also another thing, I mean, lots of very expensive, high throughput experimentation equipment is distributed in various places, and could it be better used and shared, right? So there are many things where we can think about how can we enable human biology interaction for various purposes, right? So this kind of all falls under this kind of notion, how can we make technology that enables really wider dissemination, enables people to use it, and what we would like to have is biological equipment that's robust, that it's accessible to non-experts, right? That is freely programmable, so you could make all sorts of experiments on there. You could put, it's versatile in biological content, so you could think of, you put all sorts of biology in there, and ideally it's affordable, low cost, and so forth. So it's kind of a wish list, right? And so in the following, I want to tell you about a few different projects that we've been working on to show you how we can get towards this goal. So the first one is a project that uses a single-celled organism Euglena. We build an interface where children, also adults, can basically manipulate these cells or interact with these cells just by with their finger by drawing light patterns on a touch screen. So here's a movie of that. So you see this touch screen, you see these cells, they're about 50 micrometer in size. And you can draw something on the touch screen which is then actually projected as light pattern onto these cells, right? And you can see how this a cell, for example, which is responding to the light, is actually trapped in this light circle, right? And so you can draw things on the screen and thereby interact. This is kind of the basic setup. So you have a projector, very normal projector, but with some different optics. You kind of project it into this microscopic scale. And then you have an eyepiece, so you can directly look at these cells, but you also have a camera. And then you have your tablet where you project both what's happening inside the chamber as well as what you draw over each other, right? So this is what the device looks like, very technologically. So giving people also an insight of what do we actually do in the lab with professional equipment. And if you look through the eyepiece, you can actually see all these cells, but you can also see what you've been drawing. Yeah?
Small kids play, how often do they look through the eyepiece and turn away from the screen?
So they did both, so I can tell you a little bit about that. So we've been using this system now a couple of times in the San Jose Tech Museum. Our long-term goal is to have a permanent installation there. And so we're kind of assessing through various iterations how would people, children, but also all ages, kind of interact with that. And here's kind of an example of this. What we basically found is that people are very naturally, and also children, drawn onto the system to draw stuff. So this is, for example, what a 4-year-old did. I mean, just coloring the whole screen, and that's great. Other people write their names, draw certain things. And what people notice rather fast is that there is some response there. And we had some things to actually draw them directly onto making them look through the eyepiece by some signs and things. But what also happens if one person is basically occupying the screen, then the other person has nothing really to do, is looking around and seeing the eyepiece and looking through it. And then it's actually nice that you see how then, for example, two children interact with each other and say, oh, I can see what you draw this, and do you see what I draw? And so you get additional interactions. You can also get adults involved by putting a sign there which explains how everything works, and you see how the adults actually start explaining to their children what's going on. So there's always a good thing, I've been told in museums, so adults want to appear knowledgeable to their kids, so you always need to give the written thing to the adults, and then they look smart for their children. So here's another experiment that a person did. You also see the timestamp. So this is like the whole thing happens over 2 minutes where the person for the first time interacted with the system. And you can see that the person actually did a real biology experiment, right? Like you see initially for the first view how the person just trapped one, but they made a little maze, right? Like as you see here, and then try to see how long does it take for the organism to escape the maze. And you can even say this is like a little microfluidic chip that person made just within in a few minutes, and were able to kind of interrogate those kind of organisms, right? And so it's really enabling by very intuitively giving people an experience in these microscopic worlds and these reactions of these organisms. Of course, you can make games of it. So this was kind of one of the initial intentions. So here's one example of such a game where, again, we have virtual objects on top of it, this little apple. And you're somehow supposed to, in this game, to draw a light such to make them swim to the apple, right? So, for example, like here, right? And so there's still a lot of questions for us, like what is good game design, both in terms of what makes a robust and fun game, but then also since we would like to have some educational value attached to it, and educational being very broadly spoken, how should we really design these games? And that's something we are working on right now. Right? Here's another game where basically have to get as many as you can inside the box. Right? How do you do it? Is you draw a big thing and then you zoom close over time. You can also do experiments. So a simple experiment, so you have 3 different colors you can choose. Which kind of color do they respond to? And of course, we fine-tuned the colors a little bit to make this more obvious, but it's true that kind of the blue wavelengths is the one that these cells usually try to avoid strong intensities. And so you can draw different things. You see how they swim through red and green, but not through blue, right? And so answer is blue. So a simple experiment understanding something about the phototactic behavior of these cells. Another thing which is a little bit more complicated experiments, and people don't get this as easily, but you can actually also probe where's the sensor inside the cell that actually detects the light. So what you notice is, or what people also notice, that whenever it hits the blue light, it starts turning around, which kind of indicates there must be something in the front of the cell. And if you're really skilled, you can actually draw over the back of the cell, and there's some lag in the projector, unfortunately, but if you draw over the back of the cell, you see the cell never responds. So you can do an experiment where you can actually do subcellular investigations of cells. Right? Here are some very preliminary kind of tests on children, what they see on the screen, kind of asking them draw these cells. And what you can see is that they certainly draw cells that have subcellular detail. If you ask the kids what, and we did this kind of a number of those in the museum, what these are, even though we had kind of signs saying euglena and things, basically no one says euglena. So that's something to work on. But what kids in general say is germs, bacteria, fishies, and mouse. So they suddenly realize it's real, it's alive. And it depends a little bit on the age, what they gravitate to. The older kids more towards germs and bacteria. So it also means they kind of realize it's something on a microscopic level. And certainly the eyepiece really helps with that. I mean, and so we think from a age group for more formal education, presumably somewhere 12, 13, 14 years is maybe where the kids really comprehended the whole system very easily, both the technology as well as the what's going on here. And this may actually align nicely with kind of biology education curriculum. So on this kind of project, so what I want to summarize here is that we have a system that's really long-term robust. Didn't talk about this, but the cells live happily in there for a couple of weeks if we treat them well. It's freely programmable, right? Like we can make many different applications. It's in the museum. It's really attractive for all age groups. 4-year-olds were staying there, and really found like the eyepiece is a really key thing. If you take the whole eyepiece away and just have a screen, many people think, oh, this is just another simulation, right? And so the education potential needs to be lifted up in full. So we now want to do studies on that more deeply and would like to have a permanent installation. So one thing that popped up both in our initial publication, also whenever we show this, maybe with 10% of people asking like, is this ethical, or what are the ethical considerations of that here? And so this then prompted me to basically go to Stanford bioethicist David Magnus and kind of discuss these issues through. Should we do this? Should we not? How should we do those? And wrote a paper about this. And so the bunch of potential objections one can have, and for example, going to these online comments that we got from our YouTube videos and so forth, and just I name them what comes up. Many of those you actually find, like if you talk about genetically modified food or stem cell research or so, many of these similar types of objections or concerns come up. So one major one is animal welfare, right? Like, I mean, do those organisms feel pain, right? Is this respectful for life, right? Are we doing something that is unrespectful? Although respect is kind of a little bit hard to grasp what this sometimes means. Are we playing God, right? I mean, that's an objection that people are saying. Some people say, this seems yucky, right? Like, I don't like this. It doesn't look nice to me. There's a slippery slope argument, right? Today we do it with single-celled organisms, but tomorrow we do this with monkeys, maybe, right? So, trivial pursuit, obviously it costs money and time to do these things. I mean, shouldn't we as scientists do something more useful, right? Should taxpayers' money spend better on something else? Are there public safety concerns, right? Are these organisms safe? Or maybe in the future, like some pathogen escaping into the wild because one plays something like this? Game ethics, so that was something that I brought to the debate, which bioethicists normally doesn't think about. So in the whole video game community, there's also the ethics of what makes a good video game, right? And this has to fold into there, of course, as well. If you think, for example, about discovery games, what I talked about earlier, like Eterna and so forth, there's also the question if there are discoveries made and maybe there are patents evolving and things, like who owns this if many people work on that. And there are actually a number of other things. But so those are some major ones that came up. And what we then put forward in this paper is also like some main recommendations, how one should kind of draw some boundaries to kind of be within something that one can really work with. So the first one is no pain, no harm to these organisms. Of course, also to the player. And here very explicitly to address, like the organisms we use are all single-celled, they don't have any sort of pain, they're not sentient organisms, right? And on the other hand, all the stimuli we use are actually physiological ones. And these organisms live after we played with them. So I would argue we fully covered that. Another important thing is always to engage with the public and tell them what we are doing is actually has a positive intention. That's part of the reason I'm also standing here, right? I believe strongly that these kind of things we do here could have a strong impact on education, really helping us society-wide to understand what's going on in medical research, in life science research, and so forth. Right? Respect for life, that's again a little bit harder to grasp, but one of the things that came out for me here is also, if you think about a game like this, you can think about it in two ways. One thing is you say you control the organism, right? The other thing is you can say you control a stimulus and you see how the organism responds to that, which is a very kind of different way of looking at it. And it's maybe also similar like when you play with your dog, right? I mean, are you playing with your dog, like you throw the stick and you're interacting with it, or are you manipulating your dog, right? So, very subtle but important thing. And the final thing is, of course, respect the player, right? The person who plays it. Make good games that, I mean, technically are not broken in any sense, but also have a good intention. Leave the player with a good outcome, for example, have an educational value. So that's very important, but I think that puts a good kind of foundation for us to move forward with these things, and especially develop more technology within this framework, but also strive towards education and also other applications that have a good positive outcome for society. So now I want to switch gears a little bit. It's the same organism, but now it's not so much about games, but it's about how can we provide experiments over the internet, through the cloud, to school kids so they can do experiments with these simple euglena organisms. And just, I haven't mentioned this, but just to say, these organisms are used in schools widely. So you can get them from school supplies, you find them in any sort of pond, right? You can eat them or drink them. You should not, but you could, right? They're safe. And so a typical experiment in a school class is you have them under your microscope, you shine some light from one side, and you see, do they go to the light, away from the light, things like those, right? So the setup is basically, so this is a website that we've built. This is the main screen where you see a live view. This is the microscope view that you have, and this is a joystick that you can basically actuate 4 different lights. So this is a chamber has 4 LEDs, and so you can shine light from different directions, and depending from which light from which direction the light shines, the organisms are moving in one direction. Right now, the joystick is showing upwards. They swim upwards. You move this joystick downwards, and then you see how the cells move downwards. Right? And so it's basically kind of the same experiments kids do in school anyway, except now it's over a web interface, main screen for doing the experiment, and kind of a second camera shows them the actual microscope. Okay. The way it works right now is you can log on, you get like 90 seconds, and then the data is saved for you, and you're out of the lab again. You can watch what other people are doing. You get back in the queue, and can do another experiment. What's also pretty nice and fortunate is that you almost can see, I mean, you see some nice colors, and you almost can see kind of eye spots and other subcellular details here. So this is basically the setup. Again, a microscope, a simple chamber with 4 lights from the 4 directions. We have some reservoir, so the whole thing, also important, is automated enough that it can run for up to 4 weeks without you having to change much. We have an automatic valve which from time to time puts fresh organisms in, but the organisms that come out of it are also fine. Right? So what can you do with this? On one hand, you can do a qualitative experiment, as you just saw, but then you can afterwards log into a different system. So this is 4 times sped up. You can log into a different system and analyze your actual data. And so what you see here is basically by hand tracking these cells, looking back and forth, and you even have a ruler and note the scale. So this is 0.3 millimeter. So you can measure how large euglena is. You can measure how fast it moves. And so we're now in the process of actually getting this into schools and having kids playing with that. I mean, playing in the experimentation sense, and then doing some simple tracking, doing some simple measurements. And what we haven't really implemented then, but it's also can you actually then model something? Can you even use the data to make models, right? So that's the idea. And one of the major reasons is also that it's hard for many school teachers actually to have microscopes in their school, to get these organisms in, to supply all of that, and really depends on how enthusiastic the teacher is, but also how many resources the teacher has. And so we could imagine, or that's kind of one of the things we hope, that these kind of platforms, the teacher can basically have his kids log on, do their homework on, and really do biology experiments and kind of experience these things, right? So if you look at longer time scales and we don't flush, you also see, so this is kind of almost a month, you can actually also see how the population increases, how these cells grow, how they become more dense, how occasionally the population seems to partially collapse. And if you look more closely, you can even discover events, for example, like cell division. So if you look at these two, these one cell actually, and then over time you see that there are two cells in there, right? And so it's not only about like, oh, there's a light response and you move towards the light. There's much more to be discovered in there. And the more time we actually spend in the lab of making these devices robust and doing, we all kind of discover various things that we haven't noticed before. Some of it is described in the literature, other things not, right? And so this is our interactive cloud lab. It's long-term robust, it's web accessible. When I say for everyone, right now it's only accessible for those who we give a password out. And you can basically look at biophysical phenomena on various lengths and time scales, right? Like the major thing is the light response, but there may be other things to be looked at. And we now test in educational settings what will be really striking and interesting, like letting some theoretical physicists, for example, play with that, maybe they even discover something and describe it that we hadn't known before, okay? Another project also using Euglena, so you kind of notice at some point you have an organism that works well, so you stick with that, is a kit where you can basically build a setup, a simple setup with these 4 LEDs, similar like the online setup, but play through a phone interface. And the idea is twofold. One is kind of the gameplay, but the other thing is making a kit that is low cost enough that one could kind of distribute this among students, and they could also build it so they could learn things. So again, it's the same setup. Just to recall this, it's a microscope, a little chamber, the LEDs on the side, and then you have some sort of a joystick to kind of actuate the light. Right? And so now we're going to see an actual game that was programmed in the lab. So the basic idea is a 2-player soccer game. Okay. There's a virtual soccer ball, and then you see these Euglena here, and one player basically has the joystick and then turns on the light and then makes the cells swim into one direction. So right now the red player is on and is supposed to get the ball into this goal, right? After some time, basically the player switches, so this should happen in about, I don't know, 7 seconds, and then it's the blue player's turn and actually playing in the other direction. So a few things to note is you see kind of a general movement of these cells in response to the light, but again you see lots of variability. You also note what we put down here, right? Like there's a scale bar, so this gets towards the educational content, so children hopefully noticing how big things are. But there's also, as soon as a cell has basically grabbed the ball, we also measure what's the instantaneous speed. So who would have guessed how fast the Euglena moves, right? And you get this right there. And so kind of combining this playful aspect with some more kind of educational content is kind of the intention here. Another thing we did is kind of pull out the actual Euglena, which currently has the ball in big, so with the hope to actually illuminate some more subcellular detail. The cool thing about this system is basically an Android phone and a lens, a regular eyepiece, which costs $10, and kind of a lens which costs about $10. You can, with better microscopes, you can even get better resolution of that. When the game is over, you get some stats, and for example, you get like what's the maximum speed that you had during your game, right? Like, I mean, how much is 100 micrometer per second? So how much is 100 micrometer? So this is a microscope. You can swap out the little chamber. You can put a hair under it, and you can notice that your hair has about 100 micrometer in diameter. So you can notice that the single cell swims about the diameter of a hair within a second. So those are the kind of things. This is it's intended to be a constructive kit, so you can build this. So there's a few pieces made with a 3D printer, and it basically functions as a real microscope with a real eyepiece and everything. If you take the phone off, if you put the phone on, then you already have acquisition capabilities. And the main difference that makes it interactive is basically then slotting these 4 LEDs under the microscope as well, right? So it's not only supplying to child a microscope or kind of a game kit, you supply both, a microscope and a game kit, right? The electronics to get this going, basically the whole schemata is over there, so it's not that hard. So assembling this, you learn something about electronics. We developed something to actually make these microfluidic devices, which is a little bit challenging, but so we came up with something. We have double-sided tape and some pre-cut plastic things, and you can really make these kind of chambers, which are like about 100, 300 micrometer in depth, and kind of about a square centimeter in size. And you can also think about learning programming with this thing. Of course, the game that I showed you is programmed in Android language, which is a little bit harder, so it's maybe a little more advanced. But you can also think of, when you have a game like this, to actually go to Scratch or some other child-friendly languages and then program something similar in there to what you saw on there. So we're basically in the thing that we have this in a lab, and now it's a question like how to get this out, how to get this tested with children, students of all sorts of ages, and maybe also how to get it made. One thing would be an open source way where we put all the things online, but maybe there's also some distributor who would like to make those and commercialize. Would this work in any sort of educational context? So I've been teaching together with Steve Quake bioengineering undergraduate class for the past 3 years at Stanford, and the main goal there is for the students to learn everything you need to build a device in the life sciences. And the way we did this is basically like the first half of the course, the student learned all these various techniques like optics, electronics, how to CAD, how to 3D print, and so forth, how to program. In the second part of the course, the student would build a setup. Not a phone setup, a slightly different one. But what it successfully showed is that the students had fun in kind of integrating all these various technologies into one final project that revolved around games, right? And it's kind of this idea, again, like we have these robotics classes where kids learn mechatronics because they like kind of building robots. So I think we can drive this kind of similar thing here into the equivalent concept into the life science realm, right? Like building these devices in the life sciences with some sort of a playful gaming-like environment in mind. And actually, so this is Impress now, and we will basically the whole course content for this will then also be available for others to make similar courses. So what I want to summarize here is that a kit like this could be education on 2 levels. One is building and the other one is playing, right? We have this self-builder kit, already used it successfully, kind of the idea in Stanford class. And I think the idea is it could be something equivalent like Lego Mindstorm, right? Also want to point out that Mindstorms is not a term that Lego invented. It actually goes way back to the late 70s, early 80s, just as a general concept by Papert and colleagues. Just basically giving children or students a constructive toolkit that you can build things, explore, and have fun with, and thereby learn, right? And so the next steps here is testing more with children and students and making an open source kit. And the final project I would like to talk about is another cloud lab that we built on very different organisms, works on a very different time scale, and which we used also in another class that I teach. So just to kind of show you that things also generalize a little bit beyond these kind of euglena organisms. So what you see here is a petri dish. The whole movie is actually running over about a day, so it's much slower. It takes an image about every 10 minutes. And what you should note here is the yellow thing here first. This is a slime mold called Physarum. And the slime mold is a single-celled organism but has many nuclei, and it's kind of like a tubing network. And what you also see are these drops here. They are made by a robot. This is basically food solution that the organism likes and likes to follow. And so if you watch the movie a couple of times, what you should note is just wait until it has looped, that initially the organism starts exploring, right, finds the food, and then kind of goes with the food, and is still kind of exploring to the side, but primarily following the food trail. It's kind of an interesting organism also because you kind of have 2 things mixed here. One is behavior of the organism, and the other thing is kind of morphogenesis or development, right? So it's kind of interesting to note. So again, the whole experiment you see is on the course about a day, and actually petri dish is about this size, right? And so we built robots out of Lego that would do the pipetting for us with the scanner below. So you see kind of 6 dishes on there. So this particular machine can make 6 of those experiments. And then we have an interface that works on any sort of platform that you can put in your stimuli, also get data back, and then connect this to the real experiment. So just to give you an impression what the student sees, so there's a website, you can basically choose your experiment. Then you have your current experiment that you've chosen, and then you program a stimulus. For example, here it denotes that now every 10 minutes it should make another drop along this line. This gets put into a database, and then the machine actually autonomously, on the other hand, always queries whether there's something to do for the machine, and then basically executes these chemical stimuli onto these organisms, and then an image is taken every 10 minutes by the scanner, and this whole thing is put back into the database, and when the student comes back later, can look at the execution. So it is an interactive experiment in the sense that you, throughout the day or two that experiments run, you can log in multiple times and make changes to the stimulus and see how it is going. But it's not kind of the fast scale as with the Euglena, which we saw before, which was real time, right? So as I said, we built this whole thing out of Lego, which had reasons for prototyping for us in the lab, but also may actually be a good way to get this into schools or kind of robotics classes for children to build similar systems like that. And again, kind of connecting biology to these more existing kind of Lego Mindstorm activities that are out there. And so we put this in a server rack, had 3 of those. And this was important also to already get at certain scaling issues, right? So each of those machines can do 6 experiments. We had 3 of those machines. And so you can think about server farms like this for different types of experiments that are sitting somewhere and allowing researchers, also students, to do experiments online. And just basically swapping the machines out for higher throughput equipment would allow you to run other experiments, right? So from an architectural point of view, before, like with these Euglena systems, it was really like one student was logging on to one specific system at that very amount of time. Here we have all the users kind of just interacting with the database, and all these machines autonomously query whether there's something to do in the database. And what you get out of this is actually that these individual machines could execute many more experiments in parallel. And this nicely aligns with the research paradigm which is out there, like all this high throughput equipment in biology, right? And so I use this in a biophysics class that I teach, Biophysics of Multicellular Systems, last year, where the students throughout the course were doing experiments, right? And here you see some examples what students did. For example, try to see whether you can break the organism in 2 by making 2 trails. You can check, student tested if you have lots of food versus little food, whether the organism can discriminate, all these kind of things, right? And then we asked students to do something with the data, do simple data analysis, but then also come up with models that would kind of describe what they saw. And another high-level important feature is also that you can actually track what students are doing. So you need to get proper human subject approval for that. You can track what students are doing, which helps you in 2 levels. From a teacher perspective, you can actually see which of the students has problems, who not. You could intervene and help. On a higher level, if you do this with many students anonymously, then you can also actually do learning research with it, right? Like you can see maybe there are certain things that is hard for 90% of the students, and you realize how you should actually set up things differently. And just to say, these things have been done very successfully, for example, in math, where like hundreds of thousands of kids in the US have done little math tasks online, and you have very solid data on many of these things, how maybe math is learned by children, but we don't have such a good idea about how children or students go about experimentation, right? And these kind of systems could give us insight in that. And so one of the more future ideas then is, this is pure concept art, right? But can we have a side-by-side thing where a student can do experiments, but then also have a modeling environment where you kind of can make a model, for example, of a genetic network, which is maybe taking place inside the organism, and then letting the simulator run, and then comparing the 2 with each other. And so this could teach students but also could even be kind of a discovery tool, for example, for citizen science also, right? So what I want to say here is on this project is it kind of generalizes. It's not only about microswimmers, right? You can really do this with many different organisms. The cloud lab here we had is really about parallelization, and you can really think about using much more powered high-end machines to really get much more students or people on it. We used it successfully in class, and also did some learning research in a very, small form on it. And so we're right now thinking how could we scale this up, use it in a different context, maybe different biology, and could we even get true discovery being made? So 2 last slides on outlook, like also on the future. So one thing I always want to say is that games is also a technological driver, right? It's, so there are many video games out there which require really good 3D graphics, and which huge part of the population actually wanted to play, and what you needed to do is good graphic cards, right? And so what has led to is for what we call graphics processing units, like very powerful systems to make this rendering all possible. And then what we use these machines now for, or these units, is actually, for example, making molecular dynamic simulations, many other computational tasks. Right? And it's one of these things, examples where consumer desire actually drives costs of technology that in the end feeds back into research and other more serious applications, right? And so similar things could happen with these kind of games. I mean, these biotic games and things that maybe technology gets developed, which in the end somehow synergizes with devices we use for medical research, for example. Right? I mean, you can, for example, imagine on your phone right now there's nothing that measures your blood sugar and other things, but we will have those devices, right, that really make measurements about your body and things. And having on your phone games where you can play with cells and making measurements about certain biostates may synergize in certain ways with each other, right? And the other thing I want to say is in bioengineering, we're often kind of looking to what happened in computer technology or electronics, and you can argue there are certain kind of relationships, if you kind of make a crude comparison over 50-year differences. And for example, you can equate the early foundations of quantum mechanics, like the discovery of DNA structure, right? You can talk about what are the golden years where things really took off. You can also talk about discovery of the transistor, which is really led into integrated circuits, and then compare this to like these microfluidic valves, for example, right, and leading to microfluidic circuits. And one thing, of course, to put on there is like, when did games come about? And actually, people made very early on games. There's always a debate, what was the first video game and things like those. But if you just equate this roughly, we should about now making games in biology, which we do, right? So that's good. So let me summarize. First of all, I'm very fortunate to work with a bunch of very talented people in the lab who do all of these things. Not all of them work on kind of this interactive biology. Some of them work on more conventional science to understand multicellular patterns. And the main thing I want to point out is it started for me kind of with a more game, playful experience, but I think now we made much more foray into what I now call human biology interaction with much more serious applications, like how can we bring good biology experiments, for example, to every classroom, right? The platforms that we have now are getting to the point where really long-term robust, programmable, and we really have very different ways of doing that, right? Over the web, as a phone kit, maybe something like arcade style, right? And there's educational value is really in many ways there by playing, by building, by experimenting, maybe learning analytics. We can also think about other things like just being artistic, right? Not calling this a game, but making arts with it. And what we're now working on, kind of the next steps, is really how do we get wider dissemination of these devices, encouraging other people to use them, and actually some do already, make more user study. And another thing which would really be cool if you can get actual research value, could someone make a discovery on that about biology that's really novel? And how can we increase the variation in biology? Also want to mention our funding sources. NSF, MediaX, BioX, and then our collaborators in the School of Education, Paula Blickstein, Dan Schwartz, and also Ritu Das. Okay, so thank you very much for your attention.